to the trainee talk for the day. So I'd like to introduce Coby Viner, who's from Michael Hoffman's lab. And today he'll be talking about modeling methyl-sensitive transcription factor motifs.
Thanks. So just as a number of different transcription factors are known to distinguish between different unmodified bases, some transcription factors are also known to distinguish between modified versus unmodified bases. And these different DNA modifications are known to alter the in vivo behavior of a number of transcription factors. But in addition to doing that, what they can also do is result in altered binding motifs. So here you can see a set of methylated binding motifs for various transcription factors. And note that these methyl binding motifs are quite different from their unmethylated counterparts. So they can result in completely different binding preferences in transcription factors. And there are now a number of different DNA modifications that we're interested in investigating, and our focus here is on modifications of cytosine bases, and these include the well-known 5-methylcytosine as well as its oxidative products, 5-hydroxymethylcytosine, 5-formylcytosine, and 5-carboxylcytosine. These different DNA bases can now all be sequenced. The gold standard for their sequencing is various forms of bisulfite sequencing, but it's important to note that to actually assess all of these different bases, you need to run through different sequencing runs. One sequencing run per modified cytosine base that you wish to assess. So we have the standard DNA alphabet, which is what we usually use to model transcription factor binding. And this consists of the usual A, C, G, and T. What we want to do is expand this alphabet in order to account for these different modified base pairs. And so to do that, we add symbols for these different modified bases. M for 5-methylcytosine, H for 5-hydroxymethylcytosine, F for 5-formylcytosine, and C for 5-carboxylcytosine. And so you can see here that we are now altering the sequence that we read off the double-stranded helix, but what we also need to account for is the other strand. So because you have a G always complementary to the C bases, what we want to do is model that. And so to do that, we encode symbols to preserve the complementarity relationship for these modifications. And so we add 1 for the guanine that is complementary to a 5-methylcytosine, 2 for the guanine that's complementary to a 5-hydroxymethylcytosine, etc. And now what we also want to do is add codes to represent different ambiguities in the data. So one source of potential ambiguity is that if you do certain sequencing runs, like for example just conventional bisulfite sequencing, the only thing you obtain is actually either 5-methylcytosine or 5-hydroxymethylcytosine at a given position. And you don't know which. So to encode that, we introduce ambiguity codes. So this is X for either M or H, and the complement 7 for either 1 or 2. And similarly, we introduce a code for any DNA modification or an unmodified C or G. So we don't know the modified state of this base, and so in that case we say it's Z for either M, H, F, C, or big C for cytosine. And then we also have 9 for the opposite strand. So now using this new alphabet, we can take an unmodified genome sequence and use data to now encode its modified symbols across the genome. And so we're leveraging oxidative and conventional whole genome bisulfite sequencing data from the Ferguson-Smith lab, and this was done in naive mouse T cells. And using this, what we can do is modify each position for the different modifications that we're interested in. So because we have conventional and oxidative bisulfite sequencing data, we can find M's and H's genome-wide. And so at this position, for example, we might know that it's methylated, and so it's an M because it was a C. At this position, we might know that it's methylated on the opposite strand, so it gets a 1. Whereas at this position, we don't actually know if it's methylated or hydroxymethylated, so we assign it an X. And it's important to note that this genome sequence is no longer the same for any given mouse, but actually it's tissue specific, right? So in this case, because these are DNA modifications, this is specific to the naive mouse T cells that the data is collected in. And we can then look at this in a genome browser, for example, the UCSC genome browser, and plot as tracks these different modifications genome-wide. What we can also do with this data is assess the abundance for a particular mouse specimen, and we can ask how many methylated versus hydroxymethylated bases are there. And so in this example, you can see that there's the typically expected amount of your standard nucleobases, except that some portion of cytosines are methylated. And it's important to note that the total of the methylated cytosines are both here as well as on the opposite strand here. So if you want to find how many there are in total, you have to sum both of those And so then we can see that you lose roughly 2 orders of magnitude to 5-methylcytosine, and you lose another order of magnitude to 5-hydroxymethylcytosine, and you can see that we have a reasonable number of bases for which the modification state is still not fully known. So now what we want to do is actually investigate transcription factor binding. And to do this, we use the well-known position weight matrix model, here represented by sequence logos. And what we do is we take the unmodified motif, for example, for SMAD4, and we can try testing different modifications, and then look computationally at what the effect would be. So for instance, we might modify the central CPG to be methylated, and so that would look like this. Or we might modify the central CPG to be hydroxymethylated or formylated. Or we might modify all motif positions. So here we're modifying everything to 5-methylcytosine or to other modifications. And then what we also want to do is not only assess these different hypotheses, but also try to do de novo elucidation of modified motifs. And to do that, in collaboration with James and Tim Bailey, we modified the mean suite set of tools. And this is a set of tools that's widely used for motif analyses and includes tools for de novo motif elucidation, including Beam and Dream. And we've now modified all of these tools to be able to be used with a custom alphabet, and in particular, our alphabet for cytosine modifications. So we can now do de novo motif elucidation and downstream analyses like motif enrichment with Centrimo, or annotating sequence positions by finding individual motif occurrences using this set of tools. And so the main tool that we use to actually investigate the effect of transcription factor binding is Centrimo. And what Centrimo does is it assesses central enrichment of motifs. And the way this works is you have a set of ChIP-seq data, so that's represented here. This would be the ChIP-seq signal. And then you have a set of peaks represented by the gray boxes. So these are the peaks themselves. The centers of the peaks are the gray boxes, or the summits of the peaks. And the overall black boxes are the peaks themselves. And so what you do is you scan peak centers for the best matches per region. And so that's shown here. For example, we might find that this blue motif has a number of matches, this red motif has a number of matches, whereas this orange one only has a single match. And what we would then do is count the number of sequences at each position of these central peaks. So we have our 3 red matches, which are highly central. We have somewhat less central blue matches and a single orange one. So this orange one's not going to be statistically significant in this example, but the others might be. And so we would normalize those counts to estimate probabilities, smooth it, and plot it. And then we would also get a binomial test p-value. So here you can see that the red one is more central, but less significant than the blue one, which is slightly off of center. So when we apply this, we can look at a de novo analysis, and then take the de novo motif result and put it through the Centrimo analysis. And what we get is something that looks like this. So for this example, it's the CEBP beta transcription factor. And you can see here that in orange, this is the main Jasper factor, the main Jasper motif. And the blue one is a de novo unmodified motif. That was the top bit. So that motif is shown here. And you can see that the blue one is quite less enriched than the highest enriched value, which is for this red motif here, showing the modification. And then somewhat less enriched is this cyan motif, which is fully methylated at its central position. So what this shows us is that there's a selection for a methylated CEBP beta motif. And now what we want to do is we want to run this type of analysis for a large set of factors, but in order to do that, we need to figure out what threshold to use to determine these different modified bases. So when you actually get this data, you get confidence levels, and it might be like 0.2 to 0.7 or 0.7, some value between 0 and 1. And what these values represent are our degree of confidence in a modification at a specific locus. So you might get a value for every 5MC position and a value for every 5HMC position. And so we need to determine cutoffs. So to do this, we do a grid search for all values between 0 and 1, and we use different controls. So we have positive controls that prefer 5-methylcytosine, and that's ZFP57 and CEBP beta. And then we have a set of negative controls in this case, which is just CMYC, which is known to prefer unmethylated cytosine. And now what we need to do is have Centrimo directly compare modified versus unmodified motifs in a consistent way. So to do that through hypothesis testing, we need to make additional parameters more constant. And in particular, we force a constant motif width, and we also normalize Kullback-Leibler divergence or relative entropy. And then once we do that, we get outputs that look like this. And so what you're seeing here is the result of hypothesis testing different motifs in modified versus unmodified cases. So for example, this is for CMYC, and you see the unmodified standard E-box motif here. So that's CACGTG. And we're testing all the different permutations of modifications that we expect to occur. So in this case, we might say, well, what happens if on the reverse strand it's methylated? Or on the positive strand? Or what if it's methylated on both strands? And similarly with hydroxymethylation. And what we then plot is for every value of the threshold at 0.01 increments for the modification at a specific position, we plot the ratio of the output centrimo probabilities. And so we encode that as the difference of log p values. And then what you end up with is a plot like this, where any value below 0 means that there's a preference for the unmodified motif. So since all these values are below 0, it means that CMYC prefers the unmodified motif. Whereas if we had values above 0, it would mean that it preferred the modified motif. And so in this case, you can see that for a set of samples, in every case, CMYC, the preference is toward the unmethylated motif, no matter which threshold you use. And this is a result that recapitulates what's already known about CMYC. And now for ZFP57, which you'll recall was a positive control in the other direction, so we can see that we get that result here, where all the values are above 0 consistently, and as long as you stay below a 0.7 threshold, those values are quite a lot above 0, which means that there's a strong preference for these modified motifs. And then finally for CBP beta, we can see that it prefers 5-methylcytosine, with the methylated motifs generally being above 0, but that it actually does not like binding to 5-hydroxymethylcytosine, which is consistently below 0, as shown here. And so what this means is that there's a difference that we're able to detect between the methyl binding motif versus the hydroxymethylated binding motif, and that recapitulates a result that was known for this particular transcription factor. So overall, we can see that transcription factors recognize epigenetic DNA modifications. Expanded alphabet motifs can predict differential binding affinity, and Centrimo can estimate expanded alphabet motif binding affinities. So I'd like to thank my advisor, as well as our lab, and I'd also like to thank our collaborators, especially James Johnson and Timothy Bailey, who did most of the work extending the meme suite to an expanded alphabet, as well as our collaborators at the University of Cambridge and the Sanger Institute, who provided us with whole genome bisulfite sequencing data and ZFP57 ChIP-seq. And thank you all for your attention.
Questions for Coby?
What's your next step, Coby?
So the next step is going to be analyzing this for all the transcription factors we can find in encoded ChIP-seq data. And the goal there is to try and pull out transcription factors for which we know there are either methyl or hydroxymethyl binding preferences that have yet to be reported in the literature.
So I'm not sure if I got it at the beginning, but this alphabet, this is your alphabet, right? This new alphabet to describe methylation?
Yeah, it's the alphabet that we're encoding to.
So who else is using this, if anybody?
So right now it's just us.
Okay. And how do you plan to make it, apart from the publication, how do you plan to have it used widely by the genomic
Right.
community?
So one of the tools I developed is called Cytomod, and what it does is it takes an unmodified genome sequence and it transforms it into a modified genome sequence using this alphabet. And so we're going to be releasing that, and hopefully that will be made use of. And then also for the meme suite, we have custom alphabet files which define this alphabet.
And does this new form, this transformed format, is it more compressed? Does it take as much space, more space, or the genome?
So it's going to be the same sequence. It's just that we're changing certain letters.
Yeah.
So the amount of space used will be identical. Except that for every different modified data set you want to investigate, you need a new genome. So in that case, you will be duplicating those parameter sets.
It's highly compressed compared to continuous data on methylation or
methylation. Good.
Does your alphabet conflict with some of the existing alphabets?
So currently there's no conflict except for things that are widely in use but not formalized. So for example, soft repeat masking often uses lowercase letters. And so we are overriding those letters. But there are no conflicts with things that are defined by IUPAC or any formal nomenclature.
So you might want to consider eliminating the lowercase c because it's
Yeah.
confusing without the case c.
Yeah, so that's true. Unfortunately, right now the alphabet is limited to the upper and lowercase Latin alphabet and numerals, and so it's difficult because we're already starting to run out of symbols. But it is something that we were interested in doing. Thanks.